Cell Biology 101. So the cell undergoes a cycle. This you hopefully learned about in Intro to Bio course, and a critical part of this cell cycle is cell division. So the way you go from a single cell to a whole organism is that a cell divides many times. This also happens when tissue dies and needs to be replaced. This type of cell division is called mitosis, where a single cell duplicates and separates into two essentially identical daughter cells. There are several phases of the cell cycle, and in this, the DNA replicates and then splits apart. And I want you to keep in mind that when you think about this, think about the DNA replication. So you start off and you have 2 copies, you're diploid, it replicates itself, so then you have 4 copies of every chromosome, and then the cell separate, and they end up with 2 copies of the end chromosome. So let's take a look at this cycle. We're going to start here in interphase. In interphase, this is where this cell duplicates its DNA, and then it grows a little bit, and then during prophase, chromatin condenses into chromosomes, and here in prometaphase, the membranes break down, and the microtubules start to pull things apart. In metaphase, the chromosomes line up along the metaphase plate, and then in anaphase, the cell starts to pull apart and the chromosomes pull apart, and then telophase and cytokinesis is when the cell breaks apart and cleaves into 2 daughter cells. The other important kind of cell division is meiosis, where a diploid cell divides to become haploid, and this occurs in the formation of gametes. So going through this one, you start with interphase, where you have this mother cells. They have chromosome duplication here in interphase. And then this is an important stage. In prophase, the chromosomes line up and they have crossing over. And this is where genetic material is exchanged between homologous chromosomes, resulting in recombinant chromosomes, or mixes, chromosomes that are mixes. Then in metaphase, the chromosomes line up, they split apart, and then that cell breaks down in telophase into 2 cells. But it's not done yet. Then again, in prophase, there is lining up, and in metaphase, they line up, and then they pull apart in anaphase. And then in telophase 2, the cells divide, and so you end up with 4 daughter cells that are each haploid. So you start with one mother cell that has 2 sets of the chromosomes, and you end up with 4 daughter cells that have 1 set of chromosomes. If you remember back to how heredity was discovered, it was in part with the help from Mendel the monk who worked with pea plants. And based on his observations, he pronounced that there were several laws of inheritance. So the first is the law of segregation. This is where the 2 copies of a gene in a parent separate from each other during gamete production. Here you can see this illustrated in the diagram depicting meiosis. So you start with a cell where there are 2 copies of the gene, and you end up with 4 daughter cells that each only have a single copy of the gene. So that is the law of segregation. The second law is the law of independent assortment. And this is where genes for different traits will assort randomly during the formation of haploid cells. So you can see this here in metaphase during meiosis. And so you can see that when the homologous chromosomes line up, it's not like they line up together necessarily. So you don't get only orange chromosomes in this case lining up together and then only purple ones lining up together so that you would have a cell that was identical to the parent. No, but instead what happens is there's random assortment of these chromosomes, and so they independently assort, and you end up with daughter cells that are mixes of both your mother and your father, and that are not identical to either one. All right, I want you to take a moment to think about how meiosis and mitosis might differ. One of the things is that mitosis results in identical daughter cells, 2n, and meiosis, in contrast, results in daughter cells that are haploid, 1n. And meiosis also has recombination, or crossover. So that's a couple of the ways that they're different. Maybe you can think about others.